Hi, let's do a little bit with the small intestines. So, three sections, in order, the duodenum or the duodenum, however you like to pronounce it. Either way is acceptable. So the duodenum is the first section and it's relatively short. It makes that characteristic little S shape to it. And you remember where the pancreas is, it's nestled in, sort of something like that. And there's no clear distinction between, other than sort of this drop in the duodenum where the duodenum begins, but that's the middle section. And again, there's no clear mark of where the duodenum ends and the ileum begins, at least in illustrations of this sort. Histologically, there are differences, but we're not going to get into that. The terminal portion of the small end and the largest section of it in total length is the ileum. And that then drains into the large intestine through an ileocecal valve between the ileum and the cecum. Something that you probably want to note is the spelling of ileum. It sounds just like ileum of the hip bone, but ILEUM equals the small intestine. And the ILIUM is referring to the hip bone. And the iliac arteries and veins are those that are near the ileum. So those were the blood vessels. So it's the ileum, ILEUM, which is the small intestine. And the way that I remember that, this is how my brain works, how I try to remember things like this, is that the small intestine has the E in it. And this forms the lumen of the intestine. That part of the E. I don't know if that helps you, but that's how I do it. So try to focus on the spelling. The other thing that I put on the figure is practice drawing the blood flow. See if you can just zip that out. And I'm encouraging you to keep it simple, and you're doing the arterial blood flow. So if you're going to do it simple, this is how I would probably approach it. Again, it's just concepts. So here is your aorta. And you know that you need to get blood to all of the small intestines. Well, that leaves through the superior mesenteric artery. Not drawing anything else. Not drawing the celiac trunk. That's not part of the story. Just a superior mesenteric artery. And then it just gives off branches. It goes to all of the small intestines. This is probably good enough. And it comes over and supplies part of the large intestine. That works for me. So, let's move on. Some structural features of the small intestine. This is where most of the absorption takes place and digestion, and the story involves a tremendous surface area. So all of the area of the epithelium, it's enlarged in the small intestine. So there are several ways of doing that, but first let's get oriented here. Three different illustrations that I put on this slide. Let's start with the first one, the top right. This was out of a different textbook. It's kind of nice. It shows the muscular wall of the small intestine, and it shows the mucosa, and it shows the serosa. The serosa, again, being the outer epithelial covering, the mucosa being the inner epithelial lining. And if you stick with the serosa, it wraps around the intestine, and then as it leaves the intestine, it's referred to as a mesentery. And there are mesenteric arteries and veins. Branches of them, arts and veins. And that goes off and attaches to the parietal peritoneum. There are these plica, which are simply these folds. They look like sort of the internal threads of a nut. It goes over a bolt, or spiral wrappings. That increases the surface area. It's not just a smooth tube going through. And then there are lots of villi. And you can see the villi on this top right figure. They're showing them as these little teeth. Sort of look like teeth on the edge. Well, those are finger-like projections in the bottom right. Here are two villi. Villi is plural for villus. And there is the epithelial surface in little black cells all along it. And the figure on the bottom left is an actual photo micrograph of the lumen of the small intestine, and it's where they ran a scope down into the intestine and took a picture, and you're looking down into the lumen, and the shadowy area looks to me like the lumen disappears and continues on. But the point of this is that all these little fingers that you see pointing up like little fingers on a sea anemone, those are individual villi, and there are millions of them. And again, this increases the surface area because every one of those villi is lined with this epithelium. So back to the illustration on the bottom right, showing the epithelial cells. This is simple columnar epithelium, and if you look at each of those cells as they've drawn over on the right villus and enlarged that into this vertical column of cells, there's 1, 2, 3, 4, 5, 6 cells, and what you see is that the luminal membrane of those cells is wrinkled up and contains the microvilli. And that increases the surface area of each one of those little cells. And this is sometimes referred to as the brush border. So, AKA, also known as the brush border. This sort of looks like a little paintbrush. And there are brush border enzymes that are embedded in that epithelium to digest disaccharides that are coming along into monosaccharides and dipeptides into amino acids for absorption into the cells. So I think that pretty much takes care of that illustration, that slide. Again, the story here is increased surface area for absorption. Oh, one other thing to point out in this slide are the lacteals. And as drawn in this figure are the purple. So the red is a capillary network, and the purple is a lacteal, which is involved with the reabsorption and transport of certain lipids. And that takes us, I believe, to the next slide. Yeah, so we're still in the small intestine. We're talking about absorption and transport of lipids. So, on the illustration on the bottom left, again, there's a single villus. The green is the lacteal, and the lacteal is part of the lymph system. So a lacteal equals a branch of lymph vessel. So this is lymph that's flowing through there. And the idea is that fatty acids are absorbed into the cell. So they've been broken down. They move into the epithelial cells. So they're moving into these little small black ones, and then they are reassembled into triglycerides, and those are then assembled into chylomicrons, and then those are exocytosed out the other side of the cell and taken up into a lacteal. They're then transported through the lymph. They enter the venous blood and eventually work their way to the liver. And you could trace the flow of blood perhaps from, let's say, the superior, let's write this down. Let's just do a practice here for a moment. This is just a suggestion. Let's say practice tracing blood from hang on a second. We're going into the venous blood. Let's go. Well, we'll do a couple things. Practice 1. Tracing blood from superior mesenteric vein to the inferior vena cava, IVC. Let's do another tracing problem and trace a chylomicron we'll talk about those in just a moment. A chylomicron from lacteal to IVC. Don't make a big deal out of that one, but just get it up to where the lymph drains into the I better switch that now that I think about it. Let's go to the superior vena cava. Okay. Oops. So back to the lacteals and the chylomicrons. So again, triglycerides are transported into those epithelial cells. They're assembled into chylomicrons. And then those are exocytosed out the other side into the blood vessels. Into the lacteal. Well, a chylomicron is made up of lipids and proteins. So they're surrounded by an outer layer of phospholipids and proteins. So that looks like our fluid mosaic model of cell membranes. And then there's cholesterol and triglycerides inside. Okay, so that's a chylomicron. If you look at the composition in this little bar graph on the right, sort of shows a proportional mix of those four different components. Triglycerides are the tallest, or the largest. So most of a chylomicron is made up of triglyceride. There's a little bit of cholesterol, and there's a little bit of phospholipid on the outside, and then there's a tiny little bit of protein. It's mainly how we're transporting triglycerides through the lymph to the venous blood to where it will then get to the liver. Oh, okay. Let's go back to this second tracing problem. So we're going to trace the chylomicron from the lacteal to the superior vena cava. And then let's say, where do we want to go from there? Well, let's process the chylomicron. So from the superior vena cava to the liver. How would you want to go? Probably the most direct route would just be through the hepatic artery. Okay, that's it. Last little short lecture will be a few comments on the large intestine. So we'll see you soon.